The lecture today is about conventional T cells, and we will focus on activation, differentiation, and effector mechanisms. This is the first of a series of 3 lectures on T cell immunity. My name is Natalio Garbi. I work at the Department of Cellular Immunology of the Faculty of Medicine of the University of Bonn. But before you start watching these lectures, I would recommend you that you watch the short video tutorials we have prepared on basic T cell concepts. This will help you to understand better the recorded lectures on T cells. You can find this complementary material on eCampus under the Immunology 2 video lectures, Immunology 2 Part 2, and General Concepts on T cells. It's a series of 6 video tutorials that range from basic T cell populations and T cell clonal expansion. So we can start now with this recorded lecture on conventional T cells. We'll go through 4 main points: introduction to T cells, T cell receptor recognition, naive T cell activation, what we call priming, and finally T cell differentiation and effector mechanisms. So we will start first with introduction. So broadly speaking, we can classify T cells as conventional and non-conventional T cells. Conventional T cells is what it comes first to your mind when you think about T cells. These are the typical or classical T cells, meaning the CD4 positive T cells and the CD8 T cells. They are called conventional or typical or classical simply because they were the first ones to be discovered. What characterizes them is that they are restricted, or basically they recognize, is the same. They are restricted by peptide-loaded classical MHC molecules. You will see later what classical MHC molecules are. The T cell receptor these T cells bear is highly variable, and it is characterized by an alpha and a beta chain. So here you have a dendritic cell in the middle that is presenting either MHC class II molecules loaded with a peptide, or MHC class I molecules loaded with a different peptide. The MHC class II peptide complexes are recognized by CD4 T cells via the alpha-beta T cell receptor and the CD4 co-receptor binding to MHC class II. On the other hand, CD8 alpha-beta T cells, or for short, simply CD8 T cells, they carry a different T cell receptor, also alpha-beta, and this recognizes a complex between MHC class I molecules and a different peptide. Here in this case, it is the CD8 co-receptor, the molecule that binds to MHC class I. Different to this are the unconventional T cells. And this, just by comparison to the conventional T cells, they are also called atypical or non-classical T cells. Remember, this is simply because they were discovered later. They are restricted by non-peptide ligands that are bound to non-classical MHC molecules. Their T cell receptor can be either alpha-beta or gamma-delta, depending on which unconventional T cell type you are talking about. And this T cell receptor has either a limited variability or basically it is invariant, depending again on the type of unconventional T cells. So I just told you before that we would look later what a classical MHC molecule is. This is not correct. So you have seen this already in the previous lectures, and there is a reminder in these short video tutorials that I told you earlier about differences between classical and non-classical MHC molecules. Okay, so a key characteristic of conventional T cells, this can also be applied to non-conventional T cells, is that you have 2 different types of activation. The first one is what we call antigen-dependent activation. So this is really the specific function. So these conventional T cells, they will recognize a particular antigen from a pathogen because of this antigen-dependent activation. So this can be done by all conventional T cells, meaning cells that are naive, they have never seen the antigen before, effector cells during a T cell response, or the pool of memory cells that are left after the infection is cleared. They get activated via the antigen receptor, in this case the T cell receptor, and they have a strong antigen specificity. So the response is truly antigen specific. So this T cell will be activated by antigens from, let's say, influenza virus, but not by antigens from, let's say, MCV or any other virus. The second way that these T cells can be activated, it's in an antigen-independent fashion. And this is what we call innate-like function. This is relatively new in the last maybe 5-6 years. So before this, we thought that T cells could be activated only on antigen-dependent manner, but now we know that they can also be activated on an antigen-independent manner. This can only happen on already activated T cells, either effector T cells or memory conventional T cells. Yeah? So naive T cells, as far as we know, they cannot be activated in a substantial manner via antigen-independent mechanisms. So in this case, it's not the antigen receptor that gets activated, it's not the T cell receptor, otherwise it would be an antigen-dependent activation. It is via cytokine activation. There may be other mechanisms as well that we do not know, and I'm sure that there will be something coming up here. But today, so nowadays, we know that it is cytokines that activate them. For instance, IL-18 and IL-12 produced by macrophages or monocytes will trigger this memory or activated conventional T cells to produce interferon gamma. Okay? Naive cells do not have the receptors for IL-18, so therefore they will not produce interferon gamma upon stimulation with IL-18. So this means that they are not specific for antigen. So if you have a memory T cell against influenza, and now you have an infection with Salmonella, for instance, and you have a lot of IL-18 and IL-12 produced by macrophages, this influenza-specific T cell may produce interferon gamma in response to the Salmonella infection. It's not antigen-specific, it's what we call innate-like function. So innate meaning lacking specificity. Okay, so I told you that there is a series of 3 lectures, and I would like to clarify the differences and the complementarities between these lectures. So the first lecture, the one today, we will talk about conventional T cells, okay? And we will focus on the antigen-dependent activation and function of these conventional T cells. The second lecture on T cell immunity will focus on unconventional T cells, and again, we will look at the antigen-dependent activation and function of these cells. On the last lecture, we will look at the innate functions, innate activation and functions of T cells, both conventional T cells and unconventional T cells. So conventional T cells come in different flavors, yeah, so to say. So those T cells that have never encountered their cognate or their specific antigen before, so we call them that they are antigen inexperienced, we call these cells naive T cells, yeah, so they have never been activated before. Yeah, maybe in the thymus during the development, but once they go outside the thymus, they are quiescent and they have not been activated before. And there is a very low frequency of these antigen-specific T cells. We have a lot of T cells, of course, a lot of naive T cells, but of each specificity, there is a very low frequency. This is very similar to B cells, what you have seen before with Imgar-Forster. Yeah, but now it's not B cells, it's T cells, and they don't carry a B cell receptor. Of course, they carry the T cell receptor. Once they are activated, because say you have an influenza infection, so the influenza-specific T cells now they have been activated via the T cell receptor, they become what we call effector T cells. They are not naive anymore, they have become effector T cells. So these cells are generated during a T cell response. And because of the clonal expansion, you find them at a maximal frequency that you will ever find that specific specificity. Okay? So it's exactly the same as what happens with B cells after the BCR engagement. Now, once the pathogen is cleared, most of these effector T cells die, and there are a few cells that remained of that specificity, and we call them memory T cells. Yeah, again, the same as with the B cells. Yeah, they also remain memory B cells. And even though the frequency is not as high as during the effector period, they are still increased. The frequency is still increased compared to the low frequency of the naive cells. So there are two models for this differentiation. One model is linear. So you have a naive T cell that once it gets activated MHC class 1 TCR by a dendritic cell, it will undergo clonal expansion. And after the pathogen is cleared, most of the cells will die, and the remaining ones will become memory T cells. Now, there is also other school of thought. So there are scientific evidence for both models, yeah, that they say that from the beginning you have a differentiation between effector T cells and the memory T cells. All of this, or most of this, will die after the pathogen is cleared, and the only cells that will remain will be the memory T cells. But the end result is basically the same. So where do naive T cells become activated? Well, this is a very important concept in T cell immunity that you need to understand clearly. So the naive T cells, they have a very restricted migration. Yeah, so they can recirculate only between secondary lymphoid organs, meaning spleen, lymph nodes, etc. Lymph, so they leave the lymph nodes via the lymphatics. They go back into the blood in the thoracic duct, and then from the blood they go back into the lymph nodes and back to the lymph, blood, and so on. So this is basically the life of a naive T cell. Once they are activated, so this is here in this cartoon, yeah, so a naive lymphocyte goes from the blood into the lymph node. If it doesn't get activated because there is no antigen, it will leave the lymph node via the lymph, it will go back to the blood, and so on. Now, once the T cell, the naive T cell becomes activated, yeah, it differentiates into effector T cell. And effector T cells, they basically have a virtually unrestricted migration. They basically can go wherever inflammation is taking place in the body. It can be in the skin of your toe, it can be in the skin of your ear, it can be in the liver, in the lung, wherever. And the memory T cells, there is no antigen, so in theory there is no infection, so in theory there is no inflammation unless there is a concurrent infection somewhere else, and they remain in different places. Yeah, some of them, they remain in tissues. Yeah, so we call them tissue-resident memory cells. They can provide you very fast protection and a very fast response once you get reinfected. They can also remain in the secondary lymphoid organs, or they can be recirculating like the naive T cells. Yeah, so basically patrolling the body just to make sure that once there is a reinfection, you will have a very strong response. So now we can start diving a little bit into CD4 and CD8 T cells. Yeah. So basically, the CD4 T cells, we call them the differentiated CD4 T cells. We call them helper T cells, and this is because they like helping other immune cells. Let's see how this works. So when you have a naive CD4 T cell that is primed by a dendritic cell presenting the cognate and peptide bound onto MHC class 2, this cell starts proliferating, and it will differentiate into a effector CD4 cell, which will produce cytokines and also co-stimulatory molecules. You will see later what is this. And this will lead to activation of macrophages, so they can kill the pathogens that they have phagocytosed. It can lead to activation of the B cells, as you have seen in the B cell lectures. So the B cells will be able to do isotype switch. And they can also help other cells, neutrophils, monocytes, and they can promote inflammation because they produce a lot of pro-inflammatory cytokines. On the other hand, the CD8 T cells, they also produce cytokines, so they will also help other cells, but their main function is to kill directly infected cells. Yeah? So once they are activated by MHC class 1 peptide, they will also undergo proliferation, and they become these effector CD8 cells or cytotoxic T lymphocytes because they can kill directly the infected cells. As I told you, they will also produce cytokines like interferon gamma, also produced by the CD4s, and this interferon gamma will activate macrophages and other cells as well. So you have seen here already a key point in T cell function, and it is that there is a division of labor in T cell effector function. Yeah? So you have that CD4 T cells are very good cytokine producers, and they like helping other cells to achieve their function, whereas CD8 T cells, they mainly kill those cells that are infected. Yeah? And the specific T cell activation follows recognition of the cognate MHC peptide complex. So here there is a very strong connection to antigen presentation. Yeah? So this division of labor is very much linked to the antigen presentation. Yeah? You know that CD8 T cells get activated via MHC class 1 peptide, whereas the CD4 T cells, they get activated via MHC class 2 peptide complexes. And this is very important to link now T cell activation with antigen presentation, in my opinion. So you have seen already antigen presentation with Sven Bergdorf, but I would like to just highlight a couple of things that are very important for T cell activation. So if you want to understand T cell activation, I think one of the best ways to approach this is to think of antigen presentation as a way to display the whole proteome on the cell surface of that cell. And by whole proteome, I mean the proteome of that cell, but also the surrounding extracellular space of that cell. So the T cells, when they are interacting with this antigen presentation, with this antigen presenting cell, they get a very, very well-formed opinion on what is going on that cell and on the surrounding environment via the peptide presentation. So you have here an antigen-presenting cell, for instance, a dendritic cell or an epithelial cell, doesn't matter, a cell that is presenting antigens on MHC class 1 and on MHC class 2. And you have here a CD8 T cell and a CD4 cell. So this CD8 T cell will get informed about the protein content inside this cell in the cytosol. This is because here it is one of the main proteolytic sites by the proteasome, and all proteins that are in the cytosol, being self, viral, bacterial, or tumor-derived, they will be proteolytically cleaved by the proteasome and the peptides loaded into MHC class 1 and displayed on the cell surface. On the other hand, the CD4 T cell, by interacting with this antigen-presenting cell, it gets very well informed about the protein content in the other proteolytic compartment of the cell, the phagoendolysosomal compartment, as well as the surrounding proteins that end up in these phagosomes. And this is because the proteins that are either outside the cell or in the vacuole membrane or in the plasma membrane, they end up in these endolysosomes. There they get cleaved by enzymes and into small peptides that they will be loaded into MHC class 2 molecules and displayed onto the cell surface. So as you can see, CD8 T cells, they get very good opinion and they get very good information about the protein content in the cytosol. And CD4 T cells, they get very well informed about the protein content in the vacuolar compartment and also in the extracellular medium, in the surrounding medium of these cells. Yeah? So this is how CD8s and CD4 T cells, they get different type of information conveyed by this cell. And this is important, yeah, because you see here now a dendritic cell, for instance, or a macrophage that is infected with a bacteria, say, I don't know, Salmonella or whatever bacteria. And this bacteria, this is a vacuolar bacteria that will be, of course, in vacuoles in the endosome. So therefore, MHC class 2 peptide complexes derived from this bacteria, the peptide derived from this bacteria, they will be presented into CD4 cells. The CD4 cells, there are again different flavors of these CD4 cells. In this case, it's T helper 1 and T helper 17. We will see this later. They will produce different cytokines. Th1s will produce mostly interferon gamma. And this will lead to the activation of this macrophage. Yeah? And this will lead to killing, enhanced killing of this bacteria. So now the macrophage here was not able to kill the bacteria very well after receiving the help by this CD4 Th1 via interferon gamma. It becomes very strongly bactericidal and can kill the ingested microbes because it produces more reactive oxygen species, nitric oxygen species, and so on. On the other hand, other helper cells, in this case, for instance, Th17, they don't produce interferon gamma, but they produce other cytokines, for instance, GM-CSF. yeah, or IL-17 or IL-22, yeah. And we know that some of these cytokines are very important at either recruiting other immune cells at the site of infection or activating them in situ as well. Yeah. So therefore, it will induce a strong recruitment of neutrophils to the site where there is the infection, and they will help to clear the infection. So these cells will not kill directly the infected cell, but they will help the infected cell to kill the pathogen. On the other hand, the CD8 T cells, or the cytotoxic lymphocytes, they will kill directly the infected cell. So if you have a virus or a cytosolic bacteria or a cytosolic parasite, there will be proteins derived from this pathogen that will be proteolytically cleaved by the proteasome and presented on MHC class 1. Yeah. This MHC class 1 peptide is basically invisible for CD4 cells. CD4 cells do not recognize MHC class 1, but the CD8 T cells will recognize it and will lead to the killing of this infected cell. So how do we know that T cells protect us? Yeah. So you have seen in your previous lectures that antibodies are very important to protect us against pathogens because they bind to the pathogen and neutralize it. But how do we know that T cells protect us? So all these mechanisms that I tell you, they are very nice, but what is the scientific evidence? Well, there are many different types of scientific evidence. Here I'm presenting you one in this paper. So here it is an experimental mouse model where they infect mice with Listeria, and they either leave these mice infected with Listeria and that's it. And you see, after infection, the numbers of Listeria in the spleen, they are increasing exponentially. This is a logarithmic scale. To other mice, they, in addition of infecting, they also transfer T cells that are specific for Listeria. Yeah? And now you see that these T cells are able to keep the numbers of Listeria low. Yeah? So this is very clear that the T cells are helping in controlling Listeria monocytogenes infection. However, serum, meaning antibodies, yeah, it's failing to provide immunity. So it doesn't matter whether you transfer antibodies against Listeria or not. Yeah, the number of viable Listeria in the spleen is similar. And here you have a question for you to think, and maybe some of you can give an answer when we have the question and answers session for this lecture. So why does antibody not protect against Listeria monocytogenes? Yeah, so we all know that antibodies are very important to keep us healthy and to fight infections, but why they don't protect us against Listeria? So after this introduction, we can go into the TCR recognition chapter. So one thing that I find that is really amazing is that the T cells can recognize MHC molecules bound with peptides from virtually any pathogen, and also from self-proteins, and also from food that you eat, all the protein antigens that you eat. Of course, the protein is absorbed. We need proteins, and this will also be chopped into peptides and presented into MHC and the T cells. Also inhaled proteins that we breathe in, and etc. Any protein that we have in the body. So can you imagine you have a molecule, MHC, that is able to bind so many different proteins or peptides, yeah, better say. So how is this possible in molecular terms? Yeah? So usually when if you think of MHC, this is a receptor for peptides. Yeah? And if you think of receptor-ligand pairs, usually this is highly, highly specific. The insulin receptor doesn't bind many things. It binds insulin, yeah, and so on. Okay, so how it is possible that MHC is presenting so many different peptides? Well, this is because of there are different reasons, all covered here, but we can group them in two. Yeah, one is part of the MHC, so one is focused on the MHC, and the other reason why T cells can recognize so many complexes is because of the T cell receptor itself. So let's see first the MHC. Well, the MHC repertoire, so the MHC molecules that we have, they can bind peptides from potentially any pathogen. Yeah, this is pretty amazing as well. So MHC is very exquisite for binding only peptides. This is classical MHC. They only bind peptides, but at the same time they can bind really almost an unlimited amount of peptides. Yeah, so this is a bit strange. Yeah, in molecular terms. And we can understand this very easily when we know why. And there are basically three main reasons for this. One is that MHC is polygenic. So you don't have only one MHC, one molecule, and one MHC class 2 molecule. In fact, there are three different MHC class 2 molecules coded in this case by 2 genes each because there is an alpha and a beta chain, but the resulting molecules, they are called HLA, human leukocyte antigen. This is the MHC, major histocompatibility complex molecules in humans. They are called HLA. Yeah, so you have 3 different HLA molecules, DP, DQ, and DR, and you also have 3 different MHC1 molecules in humans, HLA A, B, and C. And so you see, for class 2, you don't have only 1 molecule, you have 3, DP, DQ, and DR. The same for class 1. And then also, the second point is polymorphism. So MHC molecules, they are the most polymorphic molecules in the body. So just to give you an example, of the HLA-A gene, you have around 4,400 different polymorphisms. And most of these polymorphisms happened, not all of them, but many of them happen in the binding groove where the peptide goes. Okay, so each of these polymorphisms, so each of the polymorphisms of HLA-A will bind a different set of peptides, overlapping. Many of them will be overlapping and common for all the HLA-A alleles, but there will be also a very high number of peptides that they are different between the different alleles. Okay, so each allele will bind in the order of thousands of peptides, different peptides. And this is because they have relatively permissive peptide binding rules. Yeah, so they bind only peptides, but they have a lot of flexibility on which peptide sequence they will bind. Okay, so we can see this represented in a graphic. So this is a cell, this is MHC class 2, and this is MHC class 1. So polygenic means that you will have 3 different types of molecules for class 2 and for class 1. And polymorphic means that if you are fully heterozygous, from your mother, you will get of each a different allele to those from your father, yeah, because you are fully heterozygous. So at the end, you have 6 different MHC class 2 molecules in the surface of your cells. For MHC class 1, it's the same. You will have 1 allele, 1 polymorphic allele from your mother, and 1 polymorphic allele, a different one from your father. This is, of course, if you are fully heterozygous. Okay, so at the end, you have 6 MHC class 2 molecules and 6 MHC class 1 molecules. So therefore, you will have a lot of peptides binding to MHC class 1 and MHC class 2 because each allele binds a whole range of peptides, but also you have many different molecules, 6 for HLA class 2 and HLA class 1. Now, the T cells, they are able to recognize basically MHC loaded with antigens from basically any pathogen. Yeah, and this is also very strange because the T cell receptor has a high specificity for MHC peptide, but at the same time I'm telling you that it can be any peptide because you will always have T cells that will recognize that combination. So the influenza specific T cells that they recognize a particular MHC class 1 with a particular peptide, they will only recognize that combination. Yeah, but you have T cells in your body that will recognize basically any constellation of class 1 plus peptide. And this is because similar to the B cell receptor, you also have somatic recombination in the TCR variable regions. Yeah, so this is very similar to the B cell, to the IG locus. Of course, there are different rules, but it's very, very similar, the end result. So theoretically, you have 10 to the 15 different TCR combinations. Okay, so basically you have, in theory, 10 to the 15 different specificities. This is really a lot. Yeah, so you have a huge diversity in TCR recognition of MHC peptide. So as a result, the T cell pool can recognize virtually any MHC peptide complexes. So this means that this is a fantastic mechanism to ensure that you have specific T cells for basically any virtually any pathogen that you would encounter. But this means that each T cell has to occur in very low numbers, or each T cell specificity. Otherwise, you would not have enough space to accommodate all these T cells. So you have around each T cell carries the same T cell receptor, around 20,000 copies of the same rearranged T cell receptor. Yeah, so And theoretically, as I mentioned, you have, so there are a lot of T cell receptor molecules, but they're all the same in one cell and most likely different to the cell next to it. So that means that you have a very low frequency of these antigen-specific T cells, and it is estimated that it's around 1 in 1 million for CD4 T cells and 1 in 100,000 of CD8 T cells are specific for the same MHC peptide complex. So that means that you have to have a very strong clonal expansion, like for the B cells. So before the antigen encounter, each of these T cells with a different color, they carry different T cell receptors to each other. And this one is, for instance, specific for a particular peptide from influenza loaded on MHC class 1. Yeah, so this is 1 in 100,000. And after it gets activated, it will clonally expand. And in the infection now, you will have a very strong increase in this specificity. This depends a lot on the infection, on the specificity itself. So there is not a general rule. You can go now from this frequency to somewhere like 0.1, even 10, or even 20% at the peak of the infection. So a huge increase. So I would like to talk now a little bit on the molecular basis of this TCR recognition. Yeah? So remember, in the CD8 T cells, they recognize MHC class 1 peptide. So this is mostly derived from cytosolic sources, the peptide. I haven't mentioned in the cartoon the cross-presentation. You can go back to the cartoon, but you have seen this with Sven Burgdorf. Cross-presentation is when antigens that are in the vacuolar compartment, they end up in the MHC class 1. Okay? And you have CD4 T cells that recognize class 2 peptide, and the peptide is derived from the vacuolar and membrane antigens. Also from the cytosol via a process called autophagy. They can also end up in the MHC class 2 pathway. So if you see here, this is the top view. So if you were a T cell, this is what you could see as you are docking into an antigen presenting cell. This is the alpha 1 domain of the MHC class 1, and this is the alpha 2 domain. So these are the variable regions, and the peptide is accommodated here. And it happens that the polymorphic region of MHC class 1 is mostly concentrated in this area where the peptide binds. This is why the different polymorphic alleles bind a different set of peptides. And this is the lateral view. So here you would have the antigen presenting cell. The T cell would dock up here. And here in yellow, you have the heavy chain of MHC class 1. There is only one chain making MHC class 1 molecule. And then you have beta 2M coming from another gene, the beta 2M gene, that also binds here and gives stability to the complex. For MHC class 2, it looks similar, although it's very different in molecular terms. And you have here also, as you would dock if you were a T cell, this is the peptide, and here is the lateral side with the alpha chain of the MHC class 2 and the beta chain of the MHC class 2. And together they form here the peptide binding groove. So now if you want to look at how these two molecules interact, you have it here. It's a little bit difficult to orient yourselves. So here on the bottom, you would have the antigen presenting cell. This is MHC class 1, the heavy chain with beta 2M, and here you have the peptide. And here on top, you would have the T cell on this part here, and this is the T cell receptor, yeah? And this is the alpha chain of the T cell receptor, and this is the beta chain of the T cell receptor. And together, they form the antigen binding region. So, like in the antibody, you have constant regions called C, alpha, and beta, so they are constant, they don't recombine, and you have the variable regions where you have the somatic recombination for the rearrangement of the T cell receptor. Okay, so now we have already seen the basics of T cell receptor recognition. Now we will look at naive T cell activation, or what we call priming of naive T cells. Okay, so in the priming of naive T cells, dendritic cells play a very important role. So these are the dendritic cells are the antigen-presenting cells that are specialized to activate or prime these naive T cells. So they are much better than any other cell in the body to do this. So once the T cells have become effectors, they can be activated by any cell of the body presenting their cognate antigen. But the first initial activation, the priming, it happens most efficiently on dendritic cells. And as you have seen, naive T cells, they get activated in the secondary lymphoid organs in an antigen-specific manner. And there are there is a model that is called the 3-signal model. It doesn't happen sequentially. It probably happens almost all at the same time or in a very short period of time. And I have extended this model to include a 4th signal, the signal 0. This was discovered by Professor Christian Kurtz here at the Institute of Experimental Immunology. So in the signal 0, both the dendritic cells presenting the specific antigen and the T cells that are specific for that particular MHC peptide complex, they come together. And they do so because there are chemokines that are secreted by the dendritic cell that attract the T cells via the chemokine receptor expression on those T cells. Once they interact, there is a signal 1 that this is the antigen-specific T cell activation. Yeah? So this is when the T cell gets activated on an antigen-specific manner. At the same time, there is a signal 2 that gives a T cell survival advantage, and there is a signal 3 probably coming a little bit later when the dendritic cell is expressing cytokines that they will help in T cell differentiation. So let's see at these processes a little bit more in detail. So here you have the antigen presenting cell, in this case the dendritic cell, and here you have the T cell. So this is a CD4 T cell because here there is MHC class 2 and CD4. Yeah, and the T cell receptor. So the signal 1 is T cell receptor signaling, T cell receptor activation. Yeah, this is what gives the specificity, okay, and clonal expansion. Signal 2 typically is mediated by CD28 that recognizes B71 or CD80, and B72 or CD86. It's the same receptor. Yeah, so there are other receptors that are very important for survival. Yeah. And we will see them a bit later. IL-2 receptor, and IL-2 is one of the very important survival signals, even though it's a cytokine. Yeah? And signal 3, it's given by various cytokines that are produced by the APC or neighboring cells, immune cells or stromal cells. For instance, TGF-beta is also a signal 3 cytokine, and this is produced mostly by fibroblasts and stroma. Yeah? So this signal 3 is very important for the differentiation, whether this cell will become a Th1, a Th2, a Th17, or if it would be a CD8, whether it's going to become a strong cytotoxic T lymphocyte or a T lymphocyte, a CD8 T lymphocyte that produces more cytokines but not so much cytotoxicity. So the signal 3 is going to be very important for what you will see later, type 1, type 2, and type 3 immunity. So naive cells, I have told you that they require these 3 signals. Opposite to this are the effector T cells, so the T cells that have been already activated. Usually they are independent of signals 2 and 3, or at least they are less dependent on these signals than the naive cells. So when the activated cells get reactivated, yeah, so when they recognize now the epithelial cells infected with influenza, okay, they do not require signal 2 or signal 3. So as long as they recognize the cognate antigen, they will kill this infected cell. The same would be true for salmonella or any other antigen in the vacuolar compartment. Yeah, once the Th1s, for instance, recognize this antigen, they will produce cytokines like interferon gamma that will activate this macrophage to kill this vacuolar bacteria. For the follicular T helper cells, as you saw during the B cell lectures, they will produce cytokines that will help the B cell to induce class switch and produce IgG from IgM, for instance. Okay, so let's go through the different signals. Yeah, so we start, of course, with signal 1, TCR activation, and this is a representation of the T cell receptor and accessory molecules, as you will see. Well, the T cell receptor itself, you will be surprised, but the T cell receptor itself cannot signal because it doesn't have items. Okay, so it can get activated. There is a conformational change when it recognizes antigen, but it cannot signal itself. For this, it has complementary molecules called CD3 epsilon, gamma, zeta, epsilon, and delta. Okay, so the signal transduction of the T cell receptor requires these item motifs in the CD3, and together they form what is called as the TCR complex. Okay, So the way this works, yeah, I mean, signaling is very important. Of course, it's critical. I don't want to put too much focus on the signaling because of the time constraints. But this here represents, I mean, they have put here CD4 and CD8 together, but this you would need to see for a CD8 cell would be CD8, for a CD4 is CD4, of course. Yeah. After engagement, you have that LCK gets recruited into the CD4 or the CD8 molecule. Yeah, it's not very well known mechanistically how this works. Of course, there is a conformational change, yeah, that allows now LCK to bind, and LCK will phosphorylate the I times in the CD3 epsilon and the zeta. Now, these phosphorylated sites will recruit sub-70. It's not very clear what happens with the epsilon, but for the zeta, it's very clear that they will recruit sub-70, which in turn will phosphorylate LAT, and which in turn will allow for adaptor proteins and enzyme scaffolds to form, and you will now start driving really downstream signaling to activate PLC gamma 1, RAS, and MAP kinase pathways. As a very downstream events, yeah, so here in the middle, it's happening a lot that I will not tell you. You can look in textbook, in basic textbook knowledge. But what is very important is that downstream of these events, you have activation of certain transcription factors, particularly, but not only, NFAT, NF-kappa B, and AP1. And this will lead to proliferation of the cells, so the clonal expansion, IL-2 secretion. You remember IL-2 was a survival factor, so this will help the cell survive as well. Differentiation and effector function. Yeah, so these are three main transcription programs or transcription factors that are switched on during TCR activation. Okay, so about co-stimulation, the signal 2, as we have seen already, it promotes survival. Yeah, it's typically provided by the CD28 molecule. So this is the archetypical signal 2 that it recognizes CD80 and CD86. Also, IL-2, as I mentioned, is important, but however, there are many other co-stimulatory receptors, as you will see in the next slide. So what I would like to highlight here now is that signal 1 alone, it induces an activation of the T cells. Yeah, but signal 1 without signal 2 and without signal 3 induces what we call an abortive activation, leading to T cell anergy. So there will be a phase where the T cells will get activated, they will proliferate, these naive T cells, but they will end up in an effector stage that is not such effector. Yeah, so basically they become, so to say, lazy or anergic. If they get activated again, they will not respond. Okay, so this is a form of tolerance that you will see with Professor Veronica Lukács-Kornek in a later lecture. Signal 2 alone or signal 3 alone on naive cells, they don't do anything, or at least they don't do anything that we know. Yeah. So you really require signal 1 to start the magics on the T cell. So as I told you, there are many different co-stimulatory molecules, and some of them are listed here. So this is the antigen presenting cell, MHC, peptide, TCR, and this is the T cell. And I have told you about CD28. Another very important co-stimulatory molecule is CD40 ligand, of course. Yeah, but you have many others that they are very, very important as well. So as a kind of a balance, you also have co-inhibitory molecules. Yeah? So it's not only important to activate the T cells, it's also important to deactivate them, because otherwise could induce tissue damage by either directly killing too many cells or doing what we call bystander killing. So cells that are not present in the antigen, but they are in the inflammatory area, they are also damaged by the T cell function. And this is mediated by these co-inhibitory molecules, or probably you have heard them more about immune checkpoints. Okay, so again, TCR and MHC class 2, so here is the T cell, and you have two archetypical co-inhibitory molecules called CTLA4 and PD1. And CTLA4, interestingly, it engages the same molecules as CD28, B71 or CD80 and CD86, but the result is very different. This will activate the T cell, this will inhibit it. Yeah? And you also have PD1, you have heard a lot about anti-PD1 or PD-L1 therapy. And they promote inhibition, so you can see them a little bit as the co-stimulatory molecules are accelerators for the T cells, and co-inhibitory molecules are brakes for the T cells. So, and this is what is being exploited in tumor immunotherapy, because we now know that the T cells during some tumors, they upregulate very strongly these co-inhibitory molecules. So if you add antibodies against them to block them, then you rescue the function of these cells. Yeah. So because they express a lot of CTLA4 or PD1, they become inhibited or exhausted, they are called as well. And with these antibodies to block, excuse me, these antibodies blocking CTLA4 and PD1, they will reinvigorate the cells because they release the brake now. And finally, the signal 3 drives T cell differentiation into effector cells. And here there are several cytokines, that they drive these differentiation programs. And this we will see now in detail on the last part of this lecture, which deals on T cell differentiation and effector functions of CD4 cells, but we will also see CD8 cells later. So remember, you have seen this graphic already. It's just to repeat it and to memorize it a bit. You have a very strong division of labor in T cell function. Remember, CD4 T cells, they are called T helper cells because they help other immune cells. They help other immune effector mechanisms. For instance, they help macrophages to kill the bacteria. So they do this by secreting cytokines like interferon gamma. So this will in turn activate the macrophages that presented the antigen, increase ROS, NOS, and the bactericidal activity, and the macrophage will kill this bacteria. For the CD8 T cells, remember, they also help because they produce cytokines as well, but particularly what they do is that they are themselves cytolytic. Okay, so once they recognize the MHC class 1 peptide complex, they will produce cytokines, but they will also kill directly the cell. Yeah. Okay, and what is very important, you will see it later, the CD4 cell also helps the CD8s to get differentiated better. So it's a T helper cell. They like helping basically any cell in the immune system. Yeah. Okay, so let's see a little bit the CD4 T cell responses. Yeah. So remember the naive CD4. So this is a secondary lymphoid organ. In this case, it's a lymph node. So you will have, let's assume that you have an infection with influenza, for instance, yeah? So this would be the lung draining lymph node. So you would have dendritic cells coming from the lung into this draining lymph node, presenting antigens from influenza, because they have either been infected themselves or they have taken infected cells and present them on MHC class 1 or MHC class 2 in this case. So once they go into the lymph node, they will interact with naive cells specific for influenza that enter this lymph node, and you see here the magic starts. Signal 1 gets activated. There will be also signal 2, signal 3, and you differentiate into this helper type 1, Th1, or helper type 2, helper type 3, Th17. Okay, so clonal expansion. Now these T cells will go into the circulation, and as I told you, these effector T cells can go anywhere where there is inflammation. In this case, will be in the lung, so they will go to the lung, and when they see antigen presenting cells presenting on MHC class 2 the peptide, they will start producing cytokines that will help epithelial cells or other cells that are infected to kill the virus. Okay, via autonomous mechanisms of those cells. They will also produce chemokines and cytokines that will attract neutrophils and other cells into the site of action. So here, you've seen that there is division of labor between the CD4s and the CD8s, but also within the CD4s themselves. Yeah. So remember, the Th1s produce interferon gamma, the Th17 produce, for instance, GM-CSF or IL-17. Yeah. So this is the thymus where you have the double positive developing thymocytes, but you also have fully differentiated CD4 T cells and CD8 T cells. So the CD4 T cells, once they go to the periphery, they are basically, when they are naive, they are the same, they are not differentiated, but after they become effectors and memory cells, they will have differentiated into different flavors. So you will have Th1, Th2, Th17, and regulatory T cells. So these guys, they are very important to inhibit other T cell responses, and we will not see these cells on this lecture, but Veronika Lukács-Kornek will talk about them. And there is another subset, Th9. It's a bit controversial. People claim they are Th9. Other people claim that they are other T helper cells that they produce the cytokine IL-9. On the other hand, for the CD8s, once they go to the periphery and they get primed, they can differentiate into an effector CTL, a central memory CTL, or an effector memory CTL. So if we look at the signal 3, so the cytokines, they dictate the type of T helper differentiation that you will end up. Okay? So remember, this is a single naive cell. It recognizes this cell, this antigen by the antigen presenting cell, signal 1, and then it will start producing cytokines. Yeah, and the cytokines that this cell produces and the cytokines that this cell produces, the APC, and the surrounding cells will dictate whether this naive cell during the clonal expansion will end up being a Th2, a Th9, a regulatory, a Th17, a follicular helper cell, or a Th1. Okay, and this will the cytokines that are produced will depend very much on the type of pathogen. Okay, so if you have a lot of IL-4 production, for instance, you will get mostly Th2 responses. If you have a lot of IL-12, you will get mostly Th1 responses. Yeah, and now you have these combinations, whether you have TGF-beta with or without IL-6, different cells as well, IL-6 along with a TGF-beta, a different cell as well. Okay, so this cytokines is what is the signal 3, and each of these populations of T cells will produce different set of cytokines. And these are what we call the effector cytokines that will be produced by different transcription factors. And these transcription factors are what we call identity transcription factors. So for instance, a Th1 cell will be generated by a signal 3 of IL-12, will have the identity of a STAT4. So STAT4 will be very important for this cell to become Th1, and this is very important for production of interferon gamma. On the other hand, a Th2 will be differentiated by IL-4, will get upregulation, will express upregulated levels of STAT6 transcription factor, which is fundamental to produce the effector cytokines characterizing a Th2 response. Okay, so the effector cytokines in turn dictate the type of immune response to the pathogens that it'll be built on. Yeah. So for instance, if you have a T cell that will differentiate into a Th1 cell, this will produce interferon gamma, and it will induce a certain type of response that we call type 1. So this interferon gamma, for instance, will activate strongly macrophages and other phagocytes to kill the pathogens, and it will help B cells to switch into IgG. Okay, so as a result, the host defense is basically geared towards killing of intracellular microbes, okay, either in vacuoles or in the cytosol. Unfortunately, an excessive activation of these cells is also linked to some autoimmune diseases and tissue damage associated with chronic infections. You will see this later on in a talk by Zainab Abdullah. So if you have other cytokines, other signal 3s, that you will end up on a Th2 cell, this cell will not produce interferon gamma, or will produce very little, if any, but instead will produce other cytokines, for instance, IL-4, IL-5, and IL-13. And this will dictate a different immune response that we call type 2. And this type 2 response is very important to eliminate worm parasites, for instance, yeah, so helminthic parasites. So for these parasites, interferon gamma doesn't help much. Yeah? So it doesn't make sense to have disinfection and Th1 responses. Instead, you have these cytokines, because these cytokines will activate mast cell eosinophils and will help to activate macrophages into a tissue regeneration phenotype, because the worms make a lot of tissue damage. And all these neutrophils and tissue regenerating macrophages, they are very important for anti-helminthic defenses. Also, IgE is not very important for this type of parasites, because these cells do not express strongly the Fc receptors for IgG. They express the receptors for IgE, Yeah? And this is why the cytokines, they promote IgE formation. Unfortunately, the downside of having too strong Th2 activation is allergic diseases. They are also derived very much in a type 2 immunity. And finally, we have Th17 that they are raised by different cytokines, as we saw in the slide before, and they will produce a different set of cytokines. They produce very little interferon gamma or very little IL-13. Instead, they produce IL-17, IL-22, GM-CSF, and other cytokines. And this is very important because these cytokines either attract or activate the neutrophils. Yeah? So they bring those cells into the site of infection, and they are usually generated in cases where you have extracellular bacteria or fungi, where you need a strong neutrophilic immunity. Okay, so remember about the type 1 and type 2 immune responses. This is a very important dichotomy in T-cell or in cellular immunology. Type 1 responses are instructed by CD4 Th1 cells. Interferon gamma is a crucial effector cytokine because it activates phagocytes to kill intracellular pathogens, and it promotes the switch to IgG production. On the other hand, type 2 responses are instructed by T helper 2 cells. They produce IL-4, IL-5, and IL-13 as crucial effector cytokines, and they help to activate macrophages into a tissue regenerating phenotype. They activate mast cells and eosinophils, and they promote IgE switch to protect against helminths. Okay, so this again is the same in a different graphic representation. You have the APC presenting having a signal 1 and signal 2, and then depending on the signal 3 that is produced, because different pathogens will induce a different combination of cytokines produced, you will have either in this case upregulation of STAT4 and TBET, so you will have a type 1 immunity, or you will have upregulation of STAT6 and GATA3, leading to a Th2 type 2 immunity. So let's see how this works a bit graphically. Yeah. So you have now a dendritic cell, so you have some microbes, for instance Salmonella. Yeah, you have Salmonella, and these dendritic cells are infected, and because of the innate receptors that you saw some weeks ago, these cells will start producing IL-12. Yeah, they will not produce much IL-4. Yeah, for instance, or other cytokines. In this case, they will produce IL-12. When they activate this naive CD4 cell, it will recognize the IL-12 that is produced here because it has the IL-12 receptor. There will also be, for instance, NK cells, yeah, that because of the infection, they will produce interferon gamma. And these two cytokines will help the naive cell to become a Th1 cell. Why? Because the interferon gamma that is recognizing, it will activate STAT1, leading to expression of TBET. Remember, one of the signature transcription factors. And the IL-12 will lead to STAT4 upregulation and activation. Remember, another of the instructing transcription factors for Th1. And as a result, the cells will become Th1 cells, and they produce a lot of interferon gamma. Okay. So examples here, you have that once the Th1 is generated and it goes now Salmonella, so this was in the stomach, in the gut, yeah, so it goes to the intestine, and now it sees an APC that is infected with Salmonella. Excuse me. An APC infected with Salmonella, it presents the antigen. This APC cannot kill the Salmonella. Yeah? The Salmonella is subverting the defenses of this APC, but now with the help of interferon gamma produced by the Salmonella-specific Th1 cell, this macrophage will enhance its microbial killing ability by producing more nitric oxide, reactive oxygens, and will kill the pathogen. At the same time, this may not be enough, the interferon gamma, but now it comes an additional layer of protection. So this Th1 will help the Salmonella-specific B cells to switch into IgG. Yeah, it could be 1 or other isotypes. And now the Salmonella will be opsonized, which means that this macrophage that was also activated by interferon gamma, now it will uptake the Salmonella via the Fc receptor, and this will give a second hit of activation to the macrophage. by the Fc receptor. You saw that in the video tutorial about the B cells, yeah? And you will also have complement binding, and you will have other mechanisms that will altogether will lead to a very strong bactericidal function. So this is what we have talked about the ROS and the nitric oxide. Also, another molecule that is very important to activate the macrophages is the CD40 ligand on the T cell and the CD40 on the macrophage. Remember, this was a co-stimulatory molecule, yeah, for the T cells, but this also helps to produce interferon gamma by the effector cells, which in turn will activate these cells. So for Th2 responses, we will go also in a very similar or analogous way, so you can compare well Th1 versus Th2 differentiation. So now you have here a Helminth infection. Yeah, the dendritic cell is presenting antigens from this Helminth, yeah, and on MHC class 2. So now you have here the signal 1. And because it's a Helminth, you will have very little IL-12. Yeah. Instead, mast cells, eosinophils, and other cells that we don't really know the source, they will produce a lot of IL-4. So this is a typical cytokine that is produced under Helminth infection. And now this is the signal 3, and this will induce a different outcome on this naive T cell. So the IL-4 will generate increased STAT6 and GATA3 expression. Remember, these were the identity or the instructing transcription factors. And this will lead to expression of IL-4, IL-5, and IL-13 by these Th2 cells. And this is very important for humoral immunity. So this will induce a very important antibody response. So how does this work? Well, so again, you have the Helminth and the protein antigens from these Helminths. The proteins will be presented on MHC class 2, and now you have differentiation into Th2 and also T follicular cells, follicular helper cells. So the Th2 cells will produce IL-4 and IL-13, and this will activate the macrophages. They will not activate them to induce a lot of ROS and a lot of NOS, because this is probably not very important for Helminths, but rather it will induce a tissue repair mechanism. So you can imagine that if you have a Helminth infection, this is making holes through the organ as it moves. Some of the Helminth infections, and this needs to be repaired, yeah, and macrophages are very important for that. At the same time, the IL-5 that is produced, it will activate the eosinophils, okay? So the IL-4 and IL-13 will induce peristalsis and mucus secretion, and this is very important to discharge some of these parasites from the gastrointestinal tract, for instance. Very importantly as well, the IL-4 from the T follicular helper cells will induce switching into IgE, not IG1, not other forms of IgGs apart from the ones written here, yeah? And why is that? Why IgE is so important? Well, it is because the mast cells have mostly the Fc receptor for IgE. They don't have very strong levels of Fc receptor for IgGs, yeah? So when the antibody, the IgE antibody binds to the Helminth, this will cross-link the Fc receptors for IgE on the surface of mast cells, and this will lead to a very strong degranulation of the mast cell and toxic effect on the Helminth. So here having interferon gamma may not help a lot, yeah, and in a Th1 response, having IgE will not help a lot because mast cells don't play a role, or at least we believe that they don't play a very important role in antibacterial or antiviral immunity. So this is why you have this division of labor between different helper T cells. Okay, and finally, the development of a Th17 response. So now you say that you have bacteria or fungi that are mostly in the extracellular space. So you just have to, the cells, they just have to phagocytose these cells and destroy them. So some of these bacteria and fungi will induce IL-6 and IL-1 production. So this together, this signal 3 together with the signal 1 and TGF-beta from the surrounding cells will induce STAT3 via IL-6 or ROR gamma T via TGF-beta with the consequent release of IL-21, IL-17, IL-22 and GM-CSF, not written here, which are the effector cytokines for this Th17 response. So again, IL-22 is important for tissue repair as well. Some of these bacteria and fungi do damage, tissue damage, and IL-17 is very important to recruit and to activate other inflammatory cells. So how it would look like in the effector phase is that after activation of the naive cell, you have IL-22 production, and this is very important because it induces an increased barrier function by promoting proliferation and replacement of dead epithelial cells. So if you have a bacterial infection that is causing damage in your lamina propria, for instance, this could result in regeneration of those epithelial cells. Also, the epithelial cells will produce antimicrobial peptides in response to IL-17 or IL-22, important to kill extracellular pathogens. And IL-17 will act on tissue cells and also leukocytes to produce chemokines, TNF, and so on, which will attract further inflammatory cells into that tissue. Okay, so with this, we have done the CD4 T cells, and now we can look at differentiation of CD8 cells. So CD8 cells, remember, opposite to CD4 cells, they can directly kill the infected cells. And basically, the general process is very similar to the CD4 T cell priming. In this case, you have the dendritic cells coming from the influenza-infected lung. They go to the draining lymph node. Also, the influenza-naive CD8 T cell, the influenza-specific, will go into the lymph node via the blood. They will meet via the signal 0, you know all this, signal 1, signal 2, signal 3. Clonal expansion and differentiation, the effector T cells will live into the circulation, into the blood, and they will migrate into the infected lung, where they will kill the epithelial cells or the antigen-presenting cells that are presenting influenza antigens. So basically, how this happens, you have seen a similar cartoon earlier on. So you have an influenza virus infected dendritic cell, or cross-presenting the influenza antigens from a dead cell, that it provides the signal 1 to the T cell, signal 2 to the T cell, and the signal 3. This leads to the clonal expansion, and when these effector T cells find epithelial cell infected with influenza that presents the peptide, they will kill it. No need of signal 2 and signal 3. So if we know that this dendritic cell priming is very important for the protection against influenza, because if you have a transgenic mouse where you reduce the dendritic cells, so you can kill the dendritic cells, now you do not have cytotoxic T lymphocyte activity in vivo against influenza, because you didn't manage to expand these cells. Yeah, and if you quantify the number of cells you have with something we call tetramer, that is specific to label these specific cells, you see that if you have dendritic cells, around 15% of your CD8 cells have that specificity. Remember, 1 in 100,000, now it's 15%. But if you have killed the dendritic cells, this expansion doesn't take place so strongly. Okay, so the cytokines that are produced by the helper T cells, particularly the Th1s, they contribute very much to the priming of naive CD8 T cells. Why is this? Well, because they produce cytokines. So we can see it here graphically. So you have here an antigen-presenting cell that is presenting MHC class II peptide complexes to the helper cell, and also MHC class I peptide complexes to the CD8 T cells. So this will lead to activation of the CD4 cells, and they will produce some cytokines, like for instance IL-15, that this will help co-stimulating or giving a further signal 3 to this CD8 T cell being primed, so that it will differentiate strongly into CTL. Also, other molecules like CD40 ligand CD40 will also induce a second activation into this dendritic cell. So here the dendritic cell was activated only by the pathogen ligands. Here it is activated by the pathogen ligands, but also by this CD40 ligand interaction. Yeah. So you have here a second opinion, so to say. Yeah, the opinion that there are a bacteria or virus, and the opinion that a T cell has been activated, so it must be a strong infection and a problem we have here. And this again will lead to cytokines produced and this signal and a strong differentiation of the CTLs. So the CTLs, they, as I've told you, their effector function is to kill infected cells, and they do this by a continuous scanning. Yeah. So when this influenza-specific CD8 T cell has entered the lung, it just goes rolling and patrolling on epithelial cells. Yeah. So the first engagements are with, is non-specific, is made by adhesion molecules. In this case, LFA1 on T cells and ICAM on endothelial cells or on some epithelial cells. Yeah. So if it doesn't see the specific antigen that is presented here, because this cell is not infected, so it's not presented, yeah, then this T cell will de-engage and go to the next epithelial cell. The next epithelial cell now is different because it is expressing the MHC class 1 peptide complex that this T cell is specific for, because it is infected with influenza. Okay? And as an example. So this will lead to recognition and killing of this antigen presenting cell. So you have here some pictures like summarizing this thing. So you have here a CTL that it's not engaged with a target. So you have actin, and these granules are basically perforin and granzyme. So perforin makes holes on the target cell. T cells express the perforin, it makes holes, and the granzyme goes through these holes from the T cell into the target cell, and the granzyme is able to induce apoptosis in the target cell. And you see it here. So once you have engagement between the cytotoxic T cell and the target cell, now all the granules are polarizing on the side of the contact. You see? Here they were distributed all over the cell, but here now they are polarizing. And if you look at the electron microscope on this area here, target and T cell, you see that the T cell is secreting a lot of stuff, and some of that stuff is these granzymes and perforin, and they will create, perforin will create the pore, and the granzyme will go inside the cell to kill. Other mechanisms are also important for this death. For instance, TNF and FAS produced by the T cell are very important death-inducing mechanisms, because the target cell will express most likely TNF receptor and also FAS ligand, which is recognizing FAS. Okay. And with this, we have come up to the summary of this lecture, and it's a long list, it's a long lecture, yeah, but I've tried to summarize the most important things. Yeah? So remember, for conventional T cells, effector T cells, and memory T cells, they can be activated both in an antigen-dependent manner via the T cell receptor, but also in an antigen-independent manner via cytokines, and this we'll see on the third lecture. Yeah, so this is not signal 3, don't confuse this. Signal 3 is during naive T cell priming. This cytokine activation of effector cells happens at the effector stage. Yeah. So as I told you, the naive T cell activation is antigen dependent and is mediated by the T cell receptor. Naive cells, they express, each of them express different T cell receptors, and this means that there is a very extremely low frequency of naive antigen specific, of naive cells with that particular antigen specificity. So they require clonal expansion after signal 1 to 3. So the CD4 T cells, they become effectors and they help other cells, yeah? And they do this by promoting Ig class switch from IgM to IgG. They promote macrophage activation either into a pro-inflammatory phenotype, remember activated ROS, NOS production, or into a tissue repair phenotype. And they also help CD8 T cells to become stronger cytotoxic T cells, which become cytotoxic, as the name indicates, for infected cells. So they kill infected cells. Type 1 and type 2 immunity is regulated mostly by the helper cells. Remember Th1, Th2, Th17 for type 3. Yeah. So type 1 immunity, it's important as a mediator of cellular immunity against intracellular pathogens. Type 1 and type 3, it's cellular immunity against extracellular pathogens, and type 2 immunity promotes humoral immunity antibodies against parasites such as helminths. So I hope that you have enjoyed this lecture. I'm sure you will have a lot of questions, and some parts that they may not have been very easily understood. And I look forward very much to our question and answer sessions online, live, where we can discuss all these points. So please bring your notebook full of notes, and we'll have quite a lot of fun discussing those points. Okay, bye bye for now.